Good morning, students. Let us continue with the next topic, enzymes. We have already studied about its introduction. Now let us see what are the chemical reactions and how enzymes participate in the different chemical reactions. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compounds that undergo two types of changes. A physical change simply refers to a change in shape without breaking of bonds. This is a physical process. Another physical process is a change in state of matter when ice melts into water or when water becomes a vapor. These are also physical processes. However, when bonds are broken and new bonds are formed during transformation, this will be called a chemical reaction. So we have to study how the chemical reactions takes place by breaking the bonds and joining the new bonds. For example, barium hydroxide with sulfuric acid gives barium sulfate and water is released. It is an inorganic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. Rate of physical or chemical process refers to the amount of product that is formed per unit time. It can be expressed as rate is equals to del P upon del T, that is change in product and change in time. Rate can also be called velocity if the direction is specified. Rates of physical and chemical process are influenced by temperature among other factors. A general rule of thumb is that rate doubles or decreases by half for every 10-degree change in either direction. Catalyzed reaction proceed at rates vastly higher than that of uncatalyzed one, and that is the need of catalyst. When enzyme catalyzed reaction are observed, the rate would be vastly higher than the same but uncatalyzed reaction. For example, carbon dioxide plus water in presence of carbonic anhydrase is double as when carbonic acid breaks down into water and carbon dioxide. In the absence of any enzyme, this reaction is very slow, with about 200 molecules of hydrogen carbonate, that is carbonic acid, being formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speeds dramatically with about 6 lakh molecules being formed every second. The enzyme has accelerated the reaction rate by about 10 million times. The power of enzyme is incredible indeed. There are thousands of types of enzymes, each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction, when each of the steps is catalyzed by the same enzyme, complex or different enzymes, is called a metabolic pathway. For example, pyruvic acid is formed by the breakdown of glucose. It seems to be a one-step procedure, but actually it is a very long process, a long metabolic pathway that is having 10 steps, which all are catalyzed by enzymes. When we study respiration in chapter 14, we will study these reactions. At this stage, we should know that this is very metabolic pathway with one or two additional reactions give rise to a variety of metabolic end products. In our skeletal muscle, under anaerobic condition, lactic acid is formed. Under normal aerobic condition, pyruvic acid is formed. In yeast, during fermentation, same pathway leads to the production of ethanol, that is an alcohol. Hence, in different conditions, different products are possible with different enzymes from the same substrate. How do enzymes bring about such high rates of chemical conversion? To understand this, we should study enzymes a little more. We have already understood the idea of an active site. The chemical or metabolic conversion refers to a reaction. The chemical which is converted into a product is called a substrate. Hence, enzymes, that is protein with three-dimensional structure, including an active site, convert substrate into product P. Symbolically, this can be depicted as S converted into P. It is now understood that substrate S has to bind with the enzyme at its active site within the given cleft or pocket. The substrate has to diffuse towards the active site. There is thus an obligatory formation of an ES complex. E stands for enzyme, S for substrate. This complex is a transient phenomenon. During the state where substrate is bound to the enzyme active site, a new structure of the substrate called transition state is formed. Very soon after expected bond breaking or making is completed, the product is released from the active site. In other words, the structure of the substrate gets transformed into the structure of product. The pathway of this transformation must go through the so-called transition state structure. This we can see in the graph, how the potential energy of the substrate changes. When it is low, the substrate formed to the product is very slow. When it is high, the transition state also high. So without activation energy, it is very low. But with activation energy, it becomes very high. There could be many more altered structural states between the stable substrate and product. Implicit in this statement is the fact that all other intermediate structural states are unstable. Stability is something related to the energy status of the molecule or the structure. Hence, when we look at this pictorially, a graph, it looks like something as shown in the figure. The y-axis represents the potential energy content and the x-axis represents the progression of the structural transformation or states through the transition state. You would notice two things: the energy level difference between substrate and product. If P is at lower than S, the reaction is exothermic. One needs not to supply energy by heating in order to form the product. However, whether it is an exothermic or spontaneous reaction or an endothermic or energy required reaction, the S has to go through a much higher energy state or transition state. The difference in the average energy content of S from that of this transition state is called activation energy. ये मतलब कि यानी कि आपको जब सबस्ट्रेट को प्रोडक्ट में कन्वर्ट करना है और वो रिएक्शन जो है स्पॉन्टेनियस है या एक्सोथर्मिक है, तो उसे आपको एनर्जी प्रोवाइड करनी पड़ती है, जिसकी वजह से वो कन्वर्जन पॉसिबल हो पाता है, एंड दैट इज नोन एज एक्टिवेशन एनर्जी. Enzymes eventually bring down this energy barrier, making the transition of S to P more easy. Enzymes की वजह से ये एनर्जी जो है, दैट इज एक्टिवेशन एनर्जी का जो वैल्यू है, अमाउंट है, वो बहुत कम हो जाता है, जो बैरियर बन जाता है S से P बनने में. सो एंजाइम्स की हेल्प लेके हम ये जो ट्रांजिशन है सबस्ट्रेट टू प्रोडक्ट, इसे और ज़्यादा इजीली कैरी फॉरवर्ड कर सकते हैं. मैकेनिज्म ऑफ एन एंजाइम एक्शन. Each enzyme E has a substrate S binding site in its molecule, so that a highly reactive enzyme substrate complex ES is produced. This complex is short-lived and dissociates into its products P and unchanged enzyme with an intermediate formation of enzyme product complex. The formation of ES complex is essential for catalysis. E plus S reversibly forms ES, which forms E plus P. Concentration of substrate. With the increase in the substrate concentration, the velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity, that is Vmax, which is not exceeded by any further rise in concentration of substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules, there are no free enzyme to bind with the additional substrate molecule. The activity of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When the binding of the chemical shuts off enzyme activity, the process is called inhibition, and the chemical is called inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as competitive inhibitor. Due to this close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind, and as a result, enzyme action declines. Example: inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in the structure. Such competitive inhibitors are often used in the control of bacterial pathogens. Classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on the type of reaction they catalyze. Enzymes are divided into 6 classes, each with 4 to 13 subclasses, and named accordingly by a 4-digit number. First is oxidoreductase dehydrogenase, enzymes which catalyze oxidation reduction reactions between two substrates, S and S prime. Example: S is reduced and S prime is oxidized, will give S oxidized plus S prime reduced. Second category, transferases, enzyme catalyzing a transfer group of G between a pair of substrate S and S prime. Example: if the G is with S plus S prime is done, then G is transferred to S prime. Next is hydrolysis, enzyme catalyzing hydrolysis of ester bond, ether bond, peptide bond, glycosidic bond, carbon-carbon bond, carbon halide, or phosphorus-nitrogen bonds. Lyases, enzymes that catalyze removal of group from substrates by mechanism other than hydrolysis, leaving a double bond in between, are called lyases. Example is given: if X group is attached to one carbon and Y to the next carbon, then XY are removed, leaving double bond between the carbon atoms. Next is isomerases, includes all the enzymes catalyzing interconversion of optical, geometric, or positional isomers. Isomers have same structural formula but different 3D structures. Ligases, enzymes catalyzing the linking together of two compounds. Example: enzymes which catalyze joining of carbon oxygen, carbon sulfur, carbon nitrogen, phosphate oxygen, etc. bonds. Cofactors, enzymes are composed of one or several polypeptide chains that we have already studied in the quaternary structure of protein. However, there are number of cases in which non-protein constituent called cofactor are bound to the enzyme to make the enzyme catalytically active. मतलब जब तक नॉन प्रोटीन स्ट्रक्चर आकर अटैच नहीं होगा कुछ प्रोटीन्स में, कुछ एंजाइम्स में, तब तक वो एक्टिव नहीं होंगे. In these instances, the protein portion of the enzyme is called apoenzyme. There are 3 kinds of cofactors may be identified as prosthetic group, coenzyme and metal ion. In this kind of enzyme जिनमें कोई ना कोई नॉन प्रोटीन कॉन्स्टिट्यूएंट चाहिए, उस पर्टिकुलर कैटेगरी ऑफ एंजाइम में एंजाइम पार्ट को एपोएंजाइम कहते हैं और कोफैक्टर 3 तरह के हैं. First, prosthetic group. They are organic compounds and are distinguished from other cofactors in that they are tightly bound to the apoenzyme. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen, heme is the prosthetic group and it is the part of the active site of the enzyme. Means prosthetic group are organic compound and they are tightly attached to the apoenzyme. Next are coenzymes. These are also organic compounds, but their association with the apoenzyme is only transient, usually occurring during the course of catalysis and then they detaches. Furthermore, coenzymes serve as cofactor in a number of different enzymes catalyzed reaction. The essential chemical component of many coenzymes are vitamin. Example: coenzyme nicotinamide, adenine dinucleotide, NAD and NADP contain vitamin niacin. A number of enzymes require metal ions for their activity, which form coordination bonds with the side chains at the active site, and at the same time from one or more coordination bonds with the substrate. Example, zinc. It is a cofactor for the proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme. This testifies that cofactors play a crucial role in catalytic activity of the enzyme. So that is why they are very important to remember. So the enzymes which have two parts, they have apoenzyme plus cofactor, and cofactor are of three types: prosthetic group, coenzyme, and metal ions.